बहुत बहुत स्वागत है आपका बायोमोलिक्यूल्स के इस डिस्कशन सीरीज में। जल्दी से रिवाइज कीजिए 9.12.4 दैट इज फैक्टर अफेक्टिंग एंजाइम एक्टिविटी। चलिए द फर्स्ट क्वेश्चन, द एक्टिविटी ऑफ एन एंजाइम कैन बी affected by a change in the condition which can alter the dash structure of the protein. The next, these include comma change in substrate concentration or binding of specific chemicals that regulate its activity. Then next, again these include temperature, pH, change in substrate concentration or binding of chemicals that regulate its activity. जल्दी से रिवाइज कीजिए टेम्परेचर एंड पीएच टॉपिक्स को। चलिए द फर्स्ट क्वेश्चन, डैश जनरली फंक्शन इन ए नैरो रेंज टेम्परेचर एंड पीएच। फिगर 9.7 जल्दी से माइंड कैप्चर कीजिए इसका। The next, each enzyme show its highest activity at a particular temperature and pH called the dash temperature and dash pH. The next, each enzyme show its highest activity at a dash temperature and pH called the optimum temperature and the optimum pH. The next, activity declines both below and above the dash value. The next, low temperature pressure the enzyme in dash state, whereas high temperature destroy enzymatic activity because proteins are denatured by heat. जल्दी से रिवाइज कीजिए माइंड कैप्चर कीजिए। फिगर 9.7, इफेक्ट ऑफ चेंज इन ए pH, B temperature and C concentration of substrate on enzyme activity. The next, low temperature preserves the enzyme in a temporarily dash state, whereas high temperature destroy enzymatic activity because proteins are denatured by heat. The next, जल्दी से रिवाइज कीजिए concentration of substrate को। चलिए द फर्स्ट क्वेश्चन, विद द इंक्रीज इन dash concentration the velocity of the enzymatic reaction rises at first. The next, the reaction ultimately reaches a dash velocity, that is Vmax, which is not exceeded by any further rise in dash of the substrate. The next, this is because the enzyme molecule are fewer than the substrate molecule, and after saturation of this molecule there are dash enzyme molecule to bind with the additional substrate molecule. Figure 9.7. The next, the activity of an enzyme is also dash to the presence of dash chemicals that binds to the enzyme. The next, when the binding of the dash of enzyme activity. The process is called a dash dash, and a dash dash is called an inhibitor. The next, when the dash dash closely resembles the substrate in its molecular structure and inhibits the velocity of the enzyme, it is known as competitive inhibitor. The next, due to its close dash dash with the substrate, the inhibitor competes with the dash dash for substrate binding site of the enzyme. The next, consequently, the dash dash cannot bind, and as a result, the enzyme action declines. Example: inhibition of succinic dehydrogenase by malonate, which closely resembles the dash dash in structure. The next, such competitive inhibitors are often used in the control of dash dash pathogens. The next, जल्दी से रिवाइज कीजिए 9.1 to 5 classification and nomenclature of enzymes. The first question, thousands of dash dash have been discovered, isolated and studied. The next, most of these enzymes have been classified into different groups based on the type of the dash dash they catalyze. The next, most of these enzymes have been classified into different groups based on the type of reaction they catalyze. The next, enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by a 4-digit number dash dash. Enzyme which catalyze dash dash between two substrate S and S dash. Example: S reduced plus S dash oxidized gives S oxidized plus S dash reduced. Transferase. बताइए ट्रांसफेरेज के बारे में पूरा होल एनसीईआरटी। हाइड्रोलिसिस के बारे में, लाइएजेस, आइसोमेरेज, लाइगेज, चलिए द नेक्स्ट टॉपिक 9.1.6 यानी को फैक्टर्स को जल्दी से रिवाइज कीजिए। The first question, dash dash are composed of one or several peptide chains. The next, however, there are a number of classes in which non-protein constituents called dash dash are bound to the enzyme to make the enzyme catalytically active. The next, in this instance, the dash dash of the enzymes is called the apoenzyme. The next, 3 kinds of dash dash may be identified: dash dash group, dash dash and dash dash ions. The next, dash dash groups are organic compound and are distinguished from other cofactors in that they are tightly bound to the dash dash. The next, for example, in dash dash and dash dash, which catalyze the breakdown of dash dash to water and oxygen. Heme is the prosthetic group and it is a part of the dash dash site of the enzyme. The next, dash dash enzymes are also organic compounds, but their association with the dash dash is only transient, usually occurring during the course of dash dash. The next, furthermore, dash dash serve as cofactor in a number of different enzyme catalyzed reactions. The next, the essential chemical component of many dash dash are vitamins. Example, coenzyme dash dash, that is NAD and NADP, contain the vitamin niacin. The next, a number of enzymes require metal ion for their activity, which form coordination bonds with side chain at the active site and at the dash dash form one or more coordination bonds with the substrate. Example, dash dash is a cofactor for the pro dash dash. The next, catalytic activity is when the dash dash is removed from the enzyme, which dash dash that they play a crucial role in the dash dash activity of the enzyme. जल्दी से रिवाइज कीजिए समरी को. चलिए, the first question. Although there is dash dash diversity of living organisms, their chemical composition and metabolic reaction appear to be remarkably similar. Then next, the dash dash composition of living tissue and non-living matter appear also to be similar when analyzed dash dash. The next, however, a closer examination reveals that the dash dash of carbon, hydrogen and oxygen is higher in a living system when compared to inanimate matter. The most abundant chemical in the living organism is dash dash. The next, however, dash dash reveals that the relative abundance of carbon, hydrogen and oxygen is higher in dash dash system when compared to dash dash matter. The next, the most abundant chemical in the living organism is dash dash. The next, there are thousands of small molecular weight dash dash biomolecules. The next, amino acid, monosaccharide and disaccharide sugars, fatty acid dash dash, comma dash dash and dash dash bases are some of the organic compounds seen in living organisms. The next, there are 20 types of amino acid and 5 types of dash dash. The next, dash dash and OS are glycerides in which dash dash acids are esterified to glycerol. The next, phospholipids contain in addition a dash dash nitrogenous compound. The next, only 3 types of dash dash, that is proteins, nucleic acid and polysaccharides, are found in living systems. The next, lipids, because of their association with dash dash, separate in a macromolecular fraction. The next, biomolecules are dash dash. The next, they are made of dash dash which are different. The next, dash dash are heteropolymers made of amino acids. The next, dash dash are composed of nucleotides. The next, biomacromolecules have a dash dash of structures: primary, secondary, tertiary, and quaternary. The next, dash dash acids serve as genetic material. The next, dash dash are components of cell wall in plants, fungi, and also of the exoskeleton of the dash dash. The next, they also are storage forms of dash dash, example starch and glycogen. The next, proteins serve as a variety of cellular functions. The next, many of them are dash dash, some are antibodies, some are receptors, some are hormones, and some others are structural proteins. The next, collagen is the most abundant protein in animal world, and dash dash, that is ribosco, is the dash dash in the whole of the biosphere. The next, enzymes are proteins which dash dash biochemical reaction in the cells. The next, ribozyme are dash dash acid with catalytic power. The next, dash dash enzymes exhibit substrate specificity, require optimum temperature and pH for maximal activity. The next, they are dash dash at high temperatures. The next, enzymes lower dash dash energy of reactions and enhance greatly the rate of the reaction. The next, dash dash acids carry hereditary information and are passed on from parental generation to progeny. You are most welcome in the next session of exercise discussion. मैं उसमें आपको क्वेश्चन बोल दूंगा, आपको उसका क्वेश्चन बना लेना है। एक क्वेश्चन के बाद पॉज करना है वीडियो को और उसके बाद क्या करना है, उस क्वेश्चन को बनाना है। थैंक यू सो मच। प्लीज़ लाइक, शेयर एंड सब्सक्राइब।